Hopefully everyone can hear me. So, as Nikolai mentioned, then the main subject of this workshop really is how to find an Arabidopsis gene in wheat. So just to use as an example, we've chosen an Arabidopsis gene which is granule-bound starch synthase 1, just as a case study. And you're probably familiar with the TARE interface, so you can look for this particular gene. It's granule-bound starch synthase 1, and you have the Arabidopsis gene name, so AT chromosome 1, and then the gene locus number 32,900. And if you search with this name in Ensembl, then you can also find very similar information to what's available in TARE. And you can see here that, again, the gene is annotated as granule-bound starch synthase 1. And on Ensembl, you have different tabs which provide different information. So at the moment we're on the gene tab, but there's also a transcript tab and a location tab. On the gene tab, if you look in the left-hand side box, you have a particular set of information that's available, and the set of information that's available changes depending on whether you're in the gene tab, the transcript tab, or in the location tab. So here on the left we have things like gene trees, so how this gene relates to other starch synthase genes in other species. We also have gene ontology terms that give you some idea about the function of this gene. If we look at the transcript tab, you can see that the box on the left has changed, and we have a different set of information available. So we have sequence information. We also have information about the protein domain, so this might give you more clues about what the functions of this gene might be. And if we go to the third tab, which is the location tab, we can see where our gene is in a genomic context. So here you have GBSS1 right in the center, and you can see all the other genes that are located near to GBSS1 on the chromosome. So going back to the gene tab, we can look on the left and we can use this option called the gene tree, and this will tell us which other genes is this Arabidopsis gene related to. So looking at the gene tree, you see in red GBSS1 in Arabidopsis. And we can look and see that this other clade is the Poaceae, which includes wheat and other grasses. So we want to be able to expand this clade. And if you click on particular rectangles, that will expand all the subnodes underneath this particular node. So we'll do that and we get this massive diagram which you can't read anything on. So I've highlighted a few points of interest. So this in red is the original Arabidopsis gene we searched with. We can see, as you would expect, closely related brassica genes and other dicots like soybean. We have a separate clade at the bottom, which is the monocots, which is the one we just opened. And we have things like maize, we have rice, and we also have the Triticeae, which includes wheat. And if we zoom in on the Triticeae clade, we can see that we have 3 wheat genes from the D, B, and A subgenomes, and we also have 2 wild ancestors of wheat, Arabidopsis tauschii and Triticum muratu. And the final species included here, right at the bottom, is Hordeum vulgare, which is barley. And just to say a little bit about the gene nomenclature, then in Arabidopsis you have AT1, which is the chromosome, then G, and then a number which is the gene locus. In the TGAC gene models, these numbers are a bit more lengthy, but in essence it's a similar idea. So you have tri-AE, which is Triticum aestivum. CS42 means Chinese Spring Assembly 42. The 2DL refers to the chromosome arm, so chromosome 2, the homolog is D, and L means long, of long versus short arm. TGAC version 1 is just the TGAC genome assembly. The next number, the 157939, is the scaffold number, and then the final number is the gene ID, so AA05026660. So this is how they kind of break apart into the component parts. So you can easily tell you've got the 2DL, the 2BL, and the 2AL homologs of this gene in this gene tree. So this gene tree has enabled us to go very rapidly from an Arabidopsis gene to find the 3 wheat orthologs. But one thing to say is that these gene trees can't always be perfect, so it's important to check that your gene you found, which you think is the ortholog, really is the ortholog using a reciprocal blast technique. So this would be, you do a reciprocal blast using the coding sequence of your Arabidopsis gene, and you go to rice, not directly to wheat. This is because the rice genome is much more complete, so you're more likely to find the correct ortholog than you would if you're in wheat. You take this rice gene and blast back to Arabidopsis. Once you're content that this is the correct rice ortholog from Arabidopsis, you can then take your rice gene and blast to wheat, and then back again to rice just to check it's a reciprocal relationship. So going from our Arabidopsis gene, GBSS1, we can blast the coding sequence against the coding sequence of rice. And in this case, I found 2 genes from rice which seem to be quite feasible hits to this Arabidopsis gene. The first had about 85% sequence similarity, and the second about 80% sequence similarity. But just based on this sequence similarity, I can't be sure which one is the true ortholog. So I then did a reciprocal blast, so blasted the coding sequence of rice back against the Arabidopsis coding sequence. And for the first gene, my best hit was the original Arabidopsis gene, with a sequence similarity of about 85%. But when I blasted the second rice gene against Arabidopsis, my best hits were all other genes, so not the gene I originally searched with, and the sequence similarity was lower, only about 75%. And this suggests that actually the first rice gene is the true ortholog, because we can do a reciprocal blast and get back the same original gene that we searched with. So now that we're confident that we found the correct rice ortholog, we can then use that to look into the wheat sequence. And just to reiterate, we use the rice sequence because it's more complete than wheat. So we think that if we look in rice, we should find the gene if it's there. So that gives us confidence we're really finding the correct ortholog. So taking this rice gene, which is GBSS1, the other one is another copy of this gene in rice, which is GBSS2, but we're focusing on GBSS1. So we take the rice gene and blast it to wheat, the coding sequence, and we find 3 copies in wheat, which are about 87% similar to rice. And when we blast these back to rice, then we find the same original rice gene, which gives us confidence that this really is the correct ortholog from rice to wheat, and also going all the way back to Arabidopsis. So now we're confident that our original Arabidopsis gene is the ortholog of these 3 wheat genes. So the similarity was about 86%. And just to give you kind of rules of thumb as to how much percentage identity you might expect, then the monocot dicot divergence was about 200 million years ago, and we'd expect Arabidopsis to rice to be about 75% similar to each other in the coding sequence. You'll notice above that actually in this example the similarity is a bit higher than 75%, so nearer to 85%, and that's probably because this gene is maybe a highly conserved metabolic gene, so maybe it has less divergence between the monocots and dicots. If you go from rice to wheat, like we did, then you might expect a similarity of about 80%, and in this case again we had more similar sequences, and perhaps this is because it's a conserved function in metabolism. And just to mention, the wheat genomes are about 95% similar to each other, and just to say again, if you look for Arabidopsis to wheat, it's a low similarity of about 70%, so it's really hard to tell whether the gene you get back from wheat would actually be the real ortholog of Arabidopsis. So you really need to go via rice to be sure about the ortholog. So there are some potential pitfalls in looking for your Arabidopsis gene in wheat. So you might have the case where you have a duplication in wheat. So in this example, GBSS1 in Arabidopsis is actually duplicated in monocots. So there are 2 genes in rice, which means there's 6 copies in wheat. But using the reciprocal blast allowed us to distinguish between the 2 copies. The other thing that could happen is you might have a gene loss. So say a gene is present in a multi-gene family in Arabidopsis. Some of the genes in the family might have been lost in wheat. So to tell whether the gene you're looking for is still present in wheat, it's really best to make a phylogenetic tree using the protein sequences, and then you'll be able to tell which genes belong together. And I would also include rice in that tree to help as a kind of bridging species. The other thing to mention is that the annotations in wheat, so the gene models, are not perfect. So we tend to check that the intron-exon structure in wheat is actually conserved with rice. So when you look at the 2 gene models side by side, you'd expect to get exons which are identical in length. So in this we've highlighted in different colours each exon. So if you look, for example, the purple one is kind of the same length as the purple one in wheat, and we would expect it to be exactly the same, maybe plus or minus 3 bases, because you might get one extra amino acid. So that's something to check as well, that the gene model in wheat looks feasible. Because like Nikolai said earlier, some scaffolds are quite short, so the end of the gene might be truncated in wheat. So we've talked a lot about these TGAC gene names, but as Nikolai mentioned, there's an earlier assembly called the CSS, which is still being used at the moment because we're really in a state of transition, working with 2 different sets of gene models. And on Ensembl we have the TGAC gene models, but also on Ensembl you can find the CSS names. And when I show the expression browser, you'll see that you need to be able to use both sets of gene models really. So looking at our gene of interest, one of these GBSS1s, we can click on the settings in the gene tab and we're able to enable extra tracks. So if you click on genes and transcripts, we can turn on this extra track called alternative genes. And the alternative genes are actually the CSS genes. So once this track's enabled, we can see this new track appears called alternative genes. And here we have the original TGAC gene we looked for. And now we can see the CSS gene has been added on to the display. And just to say that the reason why we've got kind of 4 copies of the TGAC gene is because we have different transcript variants. So the gene name 0.1 means transcript variant 1. And I just wanted to also point out that sometimes you don't get an identical gene in the CSS assembly and in the TGAC assembly. So for example, this gene at the bottom here is present in the TGAC gene assembly, but not in the CSS gene assembly, and this can happen both ways around. So you have to kind of take what you know from prior knowledge and assess which gene model is best for your use. So just to summarize how to go from Arabidopsis to wheat, the gene trees available in Ensembl are really useful, but it's important to double check that you really have the correct gene using a reciprocal blast, which means you need to go from Arabidopsis to rice to wheat and back again. And once you do have a wheat gene you believe is the correct ortholog, double check that the intron exon structure looks conserved with rice, and that will confirm that the gene model is correct, just in case, for example, you're missing an exon. So if no one has any questions,